Component 3 mock, and this is paper A1, and we're up to question 4. The diagram shows a section through a red blood cell, and you've looked at these and you know all about this. And complete the table to describe and explain two different adaptations that allow the red blood cell to carry out its function. So you've really done this at GCSE, and you've done it again at A level. And these are the 4 marks that you should definitely get on this question. Some of the others are a little bit trickier, but you should be able to tell us about a red blood cell. So go for the obvious. Biconcave disc. So that's a description. It's not telling, it's not explaining it yet. And now we can go why, and we relate this to increased surface area, or possibly we could say short diffusion distance. You can see there, it reduces that diffusion distance. So we've got description, and then we've got an explanation, and then we just need another one. So for example, another typical adaptation, we've got there is no nucleus. And so if there's no nucleus, or indeed other organelles, then that leaves more room for hemoglobin. So more room for hemoglobin. And there are some other adaptations as well. You could tell us it's got hemoglobin, and the hemoglobin is adapted to carry oxygen, something like that. But these are the two, probably the two ones that you should have thought of first. B, we have got oxygen dissociation curve, and you should be happy with how that looks. The normal lung partial pressure is 10 kilopascals, and hemoglobin is 99% saturated. Well, let's just double check that, shall we? So there's my line there, and I can just my line there, and I would agree there. If that's 90, 2468, yeah, 99, bob on. What would be the effect of reducing the partial pressure of the oxygen in the lung to 8 kilopascal? So now I'm going to draw a line up from 8 and draw the line, oh, well, it's the same line across. So it's still going to be 99. So what's the effect? Well, absolutely none at all. So no change. No change at all, or maintains the same carrying capacity and so on. Part 2. Use your answer to part 1 to explain why the flat top of the curve would be advantageous to an organism. Well, this means that anywhere between 8 and 10 we're still going to get nearly maximum carrying capacity. So even if there's a small dip in oxygen content, partial pressure, then it's not going to make much of a difference. So blood can carry as much oxygen despite a small drop in the available oxygen, the partial pressure. The blood oxygenated in the lungs passes to the tissues. What would be the most likely cause of a reduction in partial pressure of oxygen in the tissues? So if we think about a muscle, and the muscle's working hard, and the oxygen is disappearing from that muscle, what explanation would we suggest? Well, respiration, aerobic respiration is going on, and that's turning the oxygen into carbon dioxide. So aerobic respiration. You could put some like exercise or something in here as well, but that's the actual process that's going on, that is being used by the tissues. The part of the curve labelled B is much steeper, so this region here. Explain how this would be advantageous to the organism. So all of these curves have got a steep part in the middle, and the steepness on here means that a small change in this partial pressure of oxygen at the bottom means a large change in the saturation. And where is this steepness? Well, most of it is actually in the tissues. So actually in the tissues, when the partial pressure goes down, then the saturation with oxygen goes down a lot. So this is another example of a question that's leading you in a certain direction. It's asking you, well, why has the oxygen gone down? And you've said respiration. And then, well, it seems to have gone down a lot. Why? Well, it's to supply it with oxygen, to unload more oxygen. So there's 2 marks here. We need to just explain the steepness and then the effect. So we say a small drop in partial pressure of oxygen a small drop in the partial pressure of oxygen, or PO2 it's sometimes called, enables a large amount or percentage of oxygen to be unloaded. I'm going to use that word, unloaded. Loading in the lungs, unloading in the tissues. So a small change in the partial pressure gives a large change in the percentage saturation of hemoglobin with oxygen. So more unloading in the tissues. I'll put in the tissues there just to make that absolutely clear. Llama hemoglobin has a higher affinity for oxygen when compared with the human hemoglobin. So you've got various examples of curves that are not fitting the standard. So llama, I always remember this, llama begins with an L, so that's going to go to the left. And there's other examples like lugworm, L, left. Fetus, well, I always think of that as little baby, L, left. And different from the other one that's going to go right, the Bohr shift. Draw a dissociation curve for the llama and label it L. Well, this dissociation curve can't go higher than 100, and it must start off at the origin. If there's no oxygen there's going to be no saturation, and it needs to be the left. So as long as it's to the left somewhere, and when it levels off, it doesn't level off higher than 100. Some that I have seen have leveled off higher than 100, but that's impossible. You can't get more than 100% saturation. So I'm just going to get that joining up there, and we're going to label that L for the mark there. Why is it necessary for llama hemoglobin to have a higher oxygen affinity? Well, that's because they live at higher altitude with less available oxygen. So the air pressure is lower, and so the fraction of oxygen in the air is also lower. So llamas live at high altitude where the availability of oxygen is lower. So there's less oxygen there. Explain what is meant by the chloride shift in the carriage of CO2. Right, so it's one of the more difficult things to remember, the chloride shift. But you've just got to see if you can blag some marks here, really, if you can't remember much. So just throw down everything that you can remember about CO2 carriage in the blood, and you'll probably hit some of these marks. So actually, 1 mark is just to do with saying this is all going on in red blood cells. So in red blood cells. So I've blagged them out there for mentioning that. And then if I actually say what happens, well, the chloride shift is the movement of chloride ions into the red blood cells. And I've blagged another mark there for not actually knowing much about the process at all, but things that I can just remember. But the full kind of story is CO2 combining with water to give carbonic acid. So our CO2 plus H2O gives our carbonic acid. And then that's combining with sodium to give sodium hydrogen carbonate. So plus sodium gives sodium hydrogen carbonate, and then this creates an imbalance, kind of electrochemical imbalance, and so that's why our chloride is being shifted into the red blood cell. So, and then we got a movement of chloride ions into the red blood cells, and that maintains and this word here, electrochemical neutrality. You can't just say makes it neutral or balances up the plus and the minuses. This is the full word that we want here. Electrochemical neutrality. So that's the kind of full story, but for 2 marks I could have said this allows, the chloride shift allows the movement of chloride ions into the red blood cells, and I'd have blagged 2 marks there. Sounds easy when you say it like that. So 13 marks in total.